So welcome, everybody. My name is Chris Penland. I work at the CF Foundation in the national office. I have the distinct pleasure of working with Catherine Tuggle and John or Jed Mahoney. And our efforts are to try to bring new therapies forward for CF patients. I just want to make sure that everybody is in the right room for this presentation. We'll be talking about efforts to characterize and correct CFTR mutations. If you were looking for information on transplant, that's going to be happening next door. Access and clinical trials is being held upstairs. So I think there was perhaps a change in the agenda. I just want to make sure everybody is where they thought they were going to be. Okay, so why don't we go ahead and get started. Catherine is going to lead us off. Catherine is the Director of Research at the CF Foundation, and she's going to talk about efforts to categorize CFTR mutations and how they are affected by different types of therapies. And then we'll transition over to Jed, who will handle the last part of the talk, and he will inform us about efforts to move what are termed induced pluripotent stem cells towards the clinic. So I want to remind everybody that at the end of the session, we will do a question and answer. Again, we're hoping that you will use your mobile device and go to cff.cnf.io to enter any questions. And I'll moderate a session after Jed presents, and hopefully we'll have a lot of insightful questions we did at the last session. So without any further ado, I'll turn it over to Catherine.
Thank you, Chris, and thank you all for being here, the very last session of the conference, so appreciate having you here. So as Chris mentioned, we're going to have the talk broken up into 3 sections. So the first part will be on the Thera Typing initiative, which was introduced yesterday by Dr. Skatch during the opening session. Then we'll shift a little more into the one-time cure, the futuristic, where we hope we're going, and then we'll follow up with questions. So as was mentioned yesterday, thanks to the work of the CFTR2 team out of Johns Hopkins University, we're now aware that there's over 1,700 mutations in the CFTR gene that are known to cause cystic fibrosis. Currently, there are 2 drugs available to patients. However, these drugs are limited in the mutations that they are currently approved for. So the Kalydeco is the first one that was approved, and it's approved for patients with 1 or more copies of 10 mutations. 2 of these mutations that are more common in the population are the G551D and the R117H. Also, Orkambi, which is a combination of ivacaftor-lumacaftor, is approved for patients with 2 copies of the F508del mutation. Between the 2 of these, we actually cover about 55% of the CF patient population with modulators. However, that means we still have 45% left to go. Keep in mind, though, I mentioned we have 1,700 mutations, and we're covering 11 right here. So we've got some work to do before we get to everybody. But I do want to draw your attention on this pie chart, this 4% right here of genotypes that we think may actually respond to some of these mutations, may respond to 1 or more of these drugs that's currently available on the market. And so the rest of my talk will really focus on how we're trying to get to that patient population. So there's a couple of ways that we can group mutations. Classically, in kind of the laboratory sense, we've really focused on using mutation classes, and this is kind of the basic mechanism, what's really going wrong in the cell, and I'll cover this in the next slide. But what we're really proposing here is to move into this Thera Typing method, where we're really looking at how individual mutations respond to therapies. And I think that's more important when we're thinking about patients because we're wanting to treat the patients. But why is this really so important? Why do we need to change the way that we're categorizing these? Well, first, we think that this may aid the design of drug trials, and this may help us to receive FDA label expansion so we can reach additional patients that we may not be able to do using the traditional types of approaches that we're using now. Additionally, kind of a longer-term goal is really to use it for personalized medicine. As we have more and more modulators become available for patients, we want to make sure that the patients are on the best drug to treat their specific mutations, and we think that some of the tools that I'll introduce today may actually help us do that. So when we talk about CFTR mutation classes, this is kind of the traditional scheme that is put up. So over on the far left, we have normal cells with the normal functioning CFTR here on the surface, and this is ideally what we want to see in everybody. However, that's not the case in CF, as patients are all affected by different mutations that have different defects. So category 1 or class 1 are mutations where no CFTR protein is made. These are typically your nonsense mutations, so a G542X would be an example of that. The class 2 mutations are mutations where the protein is made, however, it's not processed appropriately, not folded properly, and therefore it doesn't get to the surface of the cell. So it kind of gets held up here in the middle of the cell, working its way up. Some examples of mutations that fall into this class are the F508del, the A455E, and the N1303K. And I bring those up because I'll get back to them at the latter part of the presentation. Class 3 are gating mutations, so you get the protein to the surface, but it locks shut. In this, the classic example would be a G551D, which is potentiated or treated with Kalydeco. Class 4 mutations are conductance mutants, so they have the channel there, but it just doesn't open up quite all the way, resulting in a reduced amount of chloride in the exiting the cell and results in the disease phenotype. And then class 5 mutations have a low amount of CFTR on the surface, and one of the examples of this is the 3849 plus 10KB C to T. So this is how we've kind of thought about things, especially in the laboratory. We try to put them in these buckets, and we hope that it works. Realistically, though, a lot of these mutations, they fall into one category and another category, multiple defects, so it really starts to get confusing. So we've decided we need to look for something else that may be more relevant. So the concept of Thera typing is really what we're trying to understand now, if this may be something that's useful. So what is a Thera type? A Thera type is a functional grouping of mutations based on the response to a particular therapy. So an example would be if you take partial functioning mutants, gating mutants, and also some of those that have decreased expression, and they may all respond to a particular potentiator, that would be a certain Thera type. It would be that potentiator works on those mutations. Another potentiator may come down the road later, and it may not, all those mutations may not respond the exact same. There may be a little bit of a difference. So it would be drug-specific type of Thera types. Also, mutation classes may respond to multiple therapies or need multiple therapies in order to really receive clinical benefit. So some of these classes may respond to more than one Thera type. So an example would be a nonsense mutation may require a read-through agent, so you can get the protein to be made. But if that protein's also defective, you may also need a corrector and then a potentiator. So you start looking at these bigger combinations that you would need to put together in order to have a functional protein. And we think for trial design that these Thera types may be really important and maybe even better than mutation classes for trying to get mutations approved. So again, kind of what is the Thera type? This is a little bit more of a graphical representation of what I just covered on the previous slide, but to show you over here on the left-hand side. So when we're trying to look at overall CFTR function and really how you have a clinical benefit, there's two main areas or two main parameters we're looking at. It's the amount of protein you have and how well it functions. So if you look at G551D, you make a lot of the protein, however, I mentioned it locks shut. But with the addition of a potentiator, you now have a fair amount of function that has been shown in clinical studies to actually produce a clinical response. So this would be, as I mentioned, one Thera type. When we look at delta F508 over here, so with the addition of a corrector, you shift a little bit further over. You're right on that edge of a clinic where we think you might have a clinical response. And then with the addition of a potentiator, you pop right into that gray section. So again, we're seeing the clinical benefit. This is great. We have a couple of these that we've obviously proven, but we have additional mutations that we'll need to characterize in the same way to really understand what their Thera types may be. So how do we do this? Well, there's two kind of main categories that I'm going to go into to discuss today. The first is laboratory developed cell lines. These are used, they're kind of the workhorses in the lab. We use them all the time, and I'll cover those a little bit more in the next slide. The other is kind of the new, exciting part of the Thera typing initiative is really patient-derived cell models. And currently, the three main areas we're focused on are human bronchial epithelial cells. These are cells that line the airways. They can be collected either from transplanted lungs, and those have been used for traditional drug screening up until this point. But also, there are laboratories working on trying to get bronchial brushing. So if a patient goes in for bronchoscopy, they can get cells while they're down there, and we can grow those in the laboratory and really study them. Human nasal epithelial cells are a lot easier to access. You collect them with a brush. You're able to go back and get them multiple times. It's a fairly easy and almost a renewable resource that you can go back to. And then intestinal cells, these are collected from biopsies from either the large or the small intestines. For the rest of the talk, when I'm talking about personalized model systems, I'll really focus on the intestinal cells because those are a lot better developed and characterized at this point, and there's a lot of really exciting data coming out on them now. So the laboratory developed cell lines, as I mentioned, not all mutations have been characterized, so these may provide a really useful tool for understanding the basic defect with a lot of those mutations that we haven't studied. Since many mutations occur in less than 10 individuals worldwide, it's hard to access them. So again, this provides us an easy way to quickly study those mutations. So in order to generate these, we use cells that don't express CFTR and insert the different CFTR mutations into them. This provides us with a resource for really studying the mechanism and understanding how mutations may respond, or if there's mutations with multiple defects, so we can really do a lot of the basic science studying. So the picture over here is very busy, I understand, but this is data that was generated by Vertex and came out about 3 years ago, where they looked at a large number of mutations, and these are in engineered cell lines, so laboratory cell lines, and they wanted to see what the response was of specific mutations to Kalydeco, or to ivacaftor. So the dark bars at the bottom are the basic response, or the baseline response that you have with no ivacaftor. And as you can see, you have some mutations that have a lot, and you've got delta F508 over here that has nothing. You add ivacaftor, that's the white bars on top. You see some of them get even bigger, which is great, but again, down here, delta F, you have nothing, hence why that's not approved for this drug. However, you do have mutations such as R117H, where they looked at this type of data and said, wow, we think that this may actually work. So they designed a clinical trial, and subsequently that mutation was put on the label, so patients are now able to access Kalydeco. Well, this has been done for a large number of other mutations, and the Vertex 661108 study that was mentioned yesterday during the opening plenary, those are residual function mutations, and many of those are shown here in kind of the pink color. These are the ones that had some function. With the ivacaftor, they have even more function, and so we're hoping that if those trials do get, the studies from those get approved, these patients will be able to access those drugs. But these are the types of studies that are being done at the basic science level to really inform how you design those clinical trials. You wouldn't want to take a patient that's over here in this side and put them in a trial if you're expecting to see that kind of a response. So you can kind of start to discriminate. However, these tools have their limits. The single mutation may not tell you everything that's going on, so you don't understand the contribution of having that second allele in there, or other genetic and environmental modifiers, so it's important to study these in the patient as well. So with patient-derived model systems, these are really, as I mentioned, relevant to the patient. They come straight from them. And the ones that we're really focusing on at this point are easy to collect, one-time or repeated collections. And they also present with many of the complexities and variability that is observed in patients. Not all patients respond to drugs the same way, and we're hoping that some of these model systems will help us understand who would respond and who wouldn't respond and be able to tailor treatments appropriately. So just to show you over here the really cool video, these are intestinal cells that have been put into a dish, and they grow into those sphere shapes that I believe Dr. Skatch mentioned yesterday. And when you add a stimulus in there, so these are wild-type cells, so they have normal CFTR function, you see the swelling. Middle area, the chloride moves into the lumen, into the center, as you can see, small, and then it starts to balloon up. The water flows in and they swell. So these are one of the types of assays that we're really working to develop for predicting patient responses. So on this slide, we have a bit of data that was put together by the group in the Netherlands, the Jeffrey Beekman Lab, where they are starting to look at individual mutations and how they respond to these modulators. So on the left-hand side here, these are still frame images of the videos that I just showed on the previous page. So this is after they've swelled completely. So up here you have wild-type cells. You have a large lumen. This is what you want to see. With 809, 770, not really much of a difference. They're pretty much maximally swollen at this point, or stimulated. With delta 508, normally there's really no response, hence the phenotype that we see in patients. And with the addition of 809 and 770, the two components that are in Orkambi, you do start to get some swelling, so you see some effect in these cells. But what's exciting is you look down here at the A455E with the delta F508, this is another folding mutant, and you see a large lumen, so you're seeing a lot of restored function in these cells, and we think that this may be one of those mutations that may actually respond to Orkambi. So if you look over here, these are graphical representations of what I just showed. So the F508 del, baseline with 770, no response as we saw in the previous slides with the engineered cell lines. And then you have a bit of response here with the Orkambi combination. A455E with 770, you've got a good bit of response. And then with the 770 and 809, you have a huge response. However, I want to draw your attention to another class 2 mutant, another folding mutant, the N1303K. As you can see, with the addition of 770, and also with the 809, 770 combination, you don't see that type of a response. So I think this starts to show you the power of these tools to really discriminate between mutations that may respond and mutations that may not. Even though they were all in the same class, they don't respond to the same treatment options. So where do we go next? Obviously in the laboratory, we have a lot of work to do to continue to optimize and develop these protocols and really find out which systems are going to be the most useful. But also going forward, it's important to not just look at what's in the dish, but what's happening in patients. So we're coordinating with other studies that are ongoing. Some of them are observational studies funded by the foundation, but also off-label studies that are being done at a number of institutions where they're looking to see what the clinical responses are to patients where we've tested their cells. And this is just showing one example out of the Dutch group. So on the bottom, you have FEV1 increases for patients that were their organoids were tested, and then they put them on the drug to see how well they improved. And there's a nice correlation so far, granted a small number of samples, but as the FEV1 increases, they also found that there's an increase in the swelling assay. So we think that there's a lot of promise with this type of technology and some of the others. And so this is kind of the first step, but where we're hoping to go in the next year or so to really understand more and more mutations and which ones may respond. And with that, I'm going to turn it over to Jed.
All right, first off, I'd like to thank Catherine for turning this over to me. And I guess I'd like to start off by saying I'm Jed Mahoney. I work at the CFTR Lab. And how many people here knew that there was a full biological research lab going on? That's awesome. That's fantastic. That makes me excited. That makes me very, very happy. That's great. So, all right. So let's talk about stem cell biology. So I run the stem cell biology group here. And I wanted to start with this slide for two reasons in particular. Number one, if we look at the human lung, that's what this is right here. This is a cast of the airway. So this was a cadaver lung where a resin was put down the airway and then the tissue was all digested. And when you look at this lung, you can see how complex, instantly you can see the complexity of the lung. And this would just be the epithelium. This doesn't have the smooth muscle component to it. Over here in blue and red, you have the venous component to it. You don't have the innervation. So immediately you can start appreciating how complex the lung is all by itself. And so the question naturally arises to somebody like me, which says, well, how did that develop? Where did that come from? And so this right here is a snapshot along developmental time points of the mouse lung in development. So what we have here is the postnatal day 1, which is that first day after the mouse was born. And then when we back it all the way up, this is right around lung specification. And these two little buds over here on that far side, they contain some cells that have the ability to form the entire branching network of the lung. And that right there is the bona fide airway stem cells for the lung. So I'm going to do this talk a little backwards than I would normally do it. I'm going to talk to you guys about a few techniques and a few definitions, and then I'm going to tell you how we're going to be applying those in the lab. So I guess the most important one is what is a stem cell? Okay, so stem cells have two critical criteria that they have to reach to be considered a stem or progenitor cell. And that is, number one, they must be able to self-renew. So they must be able to divide and give rise to a very similar cell. And number two is that they must also be able to divide and give rise to progeny that are different, either functionally or phenotypically. So here in this little classical diagram, we have this kind of tan cell turning into a blue or the red, and then into either a lung cell here, this is a ciliated cell, or a neuron over here. And that these cells then can make up a greater part of the tissue, which can be functional. So that's really the key. But the question then comes out of, how do we use these cells for drugs? So I like this representation of stem cell biology slightly better than that very standard one. And this is what's known as Waddington's landscape. Okay? And what this does is it sets the stage that in stem cell biology, you're starting at the top of the hill, and that's your undifferentiated cell. And then depending on the factors and the environments that are influencing that cell, it'll start rolling down the hill and it will get nudged one way or another into a different one of these valleys where it will ultimately endogenously know how to become that cell of interest. So that right here would be coming into a lung here, for example. Now, recent advances have come up with this idea by Yamanaka over in Japan, which has been able to take a fully differentiated cell from blood or from skin and then reprogram it. And it can bring it all the way back to the top of the hill. So that's going back to the place where you can make any cell type in the human body. And then in vitro, or in the dish, we can apply those nudges and those environments to kind of push it which way we want. And ultimately, my goal is to produce more lung cells. So this is the material that which my lab primarily focuses and works on, which are known as induced pluripotent stem cells. Another technology that my lab relies on quite a bit is very recently discovered, very recently classified in the last 6-7 years, and that's this CRISPR and Cas technology. And what this is, it's an enzyme that was discovered in bacteria, and this enzyme was known to be able to cause double-stranded breaks in the genes. So now the beauty of this exact enzyme is we can target it, and we can target it very specifically, and that will lead to a double-stranded DNA break. So in this example that I have up here is that a W1282X gene for CFTR. We can target this enzyme to this exact allele at this exact nucleotide position, have it cause a double-stranded break. And when that double-stranded break occurs, we can influence the repair process and have the repair process remove that disease-causing nucleotide sequence and put back in a normal or a wild-type sequence, therefore almost correcting the disease in that cell. Now, the beauty of this is that if we do this in stem cell biology, we can have one dish that's uncorrected and another dish that has been corrected. And that leaves us the ability to compare down to the single base pair change what the drugs are doing in these two different populations. All right, so what does my lab want to do? This is my long-term focus right now, is a mutation agnostic, one-time cell-based therapy cure. And this is how we, thank you. Yeah, I like that. All right, so great. So this is the vision from my, I like that, thank you. All right, so this is the vision from my lab. We can find a patient that has CF early on in the disease course. We can remove some blood as a simple needle draw. We can bring that blood to the lab. We can reprogram those cells to being back to that induced pluripotent state. When they're there, we can work with them for a great amount of time. We can cause gene editing. We can correct that mutant allele, that disease-causing nucleotide sequence, direct them back to the lineage of the lung through the, pushing it down the hill in the way that we want, and then hopefully be able to transplant it back into the original patient, causing a one-time cure. Now this, I have to tell you, first and foremost, there's a lot of basic science that still needs to be going on before this is going to happen, but plausibility and feasibility of this seems to be very, very, very significant. And so we're doing that work right now in that lab up in Lexington, Massachusetts. And what we're learning is directly translating to our drug screening efforts. So right now we can take blood from a patient, reprogram them, directly differentiate them, and put them into our drug screening efforts. And you wonder, what good is that? Well, when I think about those many, many mutations out there that are very rare, we need an incredible amount of material to do any form of drug screening. And this gives us that ability. We can expand those undifferentiated cells to great amounts, and then we can process them into our drug screening assays and look for therapies that might benefit those very rare patients, giving us the biomass necessary to do the drug screen. So I'm going to give you a little example. This is work that's come out of our lab. The question is here now, how do we do this work? So we follow developmental principles. So this here is a little schematic of the mouse development. And basically over the last 40, 50 years, a lot of developmental biology has gone into identifying what signaling pathways are active where and when. And how do those relate to the cell phenotypes that come out later on down the line. So we've gone and mined all of that data, and we apply those principles in the dish. So we know what pathways get turned on at what time, and we do the same thing. So here we have a group of cells that are all entirely blue, okay? And we want to make them into these green lung cells. Well, we know which factors are active in development, so we apply those factors. And so we might have one green cell here, but then we can enrich to having these clusters of green cells here, and these clusters are very phenotypically reminiscent of these early lung buds. Then we can enrich and do our best to purify out those cells that are going to be lung lineage specific and are going to be embryonic lung-like cells. And we can get to about 70% right now efficiency in getting those cells to be very reminiscent of that lung. So what we can do with those cells as it stands today is we can put them into some basic electrophysiology assays, and we can put them into some other assays that we're working on developing right now. So these are examples of that. Using these derived cells, we've been able to do electrophysiology, which shows us that we have a pharmacology window. So this is a delta F508 patient here. It's a homozygous for delta F508. And what you see here in red is the treatment of VX809, and in black is under the vehicle control. And you can see 809 in these cells does represent the same change that it does using the primary bronchial epithelial cells. Now that's a great proof of concept for the delta F508, but we also have been able to recently, very recently, demonstrate here that this is one of those X mutations, that we can do the same thing. We can push those cells to the lung lineage. We know they express CFTR. We can put drugs into these culture conditions, and we can look for the protein. So here's a W1282X row, and you can see this is how much protein we have without any drug in our vehicle control. We have some known drugs that will modulate the X mutation, and you can see we're actually starting to show protein expression in that W1282X under these pharmacology conditions. So I consider this and the next slide to be the most important slides that are here. Basically, it's showing you a picture of the people that are there every day that are doing this work and who are really uniquely focused on getting those rare mutations classified and starting to look for those rarer disease therapies. So, awesome. So in conclusion, you know, we're kind of a ways off, and I know that's kind of frustrating to hear, but you should take home the feasibility of these studies really does seem to have consequence. We really do think that this can be a useful technique for us for the long term. But the beauty of it is, as we're pushing for that one-time cure, we're learning so much about these cells and how to apply these cells in drug screening right now. So this is not, you know, fraught. This is work that we're going to get usable therapies out of. And also, with the combination of those stem cells and gene editing, we're actually looking down to that one base pair change. So we're looking at the disease-causing element. No longer am I looking at my cells versus somebody else's cells. Now I'm looking at my cells with the disease versus my cells with the corrected disease. That removes all those modifier genes, and it gets rid of any of the confusion when it comes to patient variability. So with that, I'm going to put up this slide here. I'm going to highlight again the people that are there all day long, every day, who are focused and dedicated at doing this work, and I'm happy to take your questions. So please ask what you know, ask what you want, and we'll try our best to answer.
So, Catherine and Jed, thank you for those very nice presentations. We have some questions that have already been submitted online, and why don't we go ahead and start with you, Catherine? And I think this probably relates to an upcoming workshop that we're having. So how do we bring this laboratory research about Thera Types and apply it towards the clinic? I think you touched upon it a little bit, but perhaps tell us what's rapidly on the horizon.
Yeah, so we have a long, or kind of an initiative underway right now, but it's really starting out with a workshop in about 2 weeks. We're bringing in international experts working with these cell models. Some of the data that I showed from the Netherlands, we have the person who developed that technique. We have people around the U.S. coming in, we have clinicians, and we also have the regulators coming in to say, okay, where are we now? Where do we need to go? And how is this actually going to help us to get through the regulatory process? Because we see, you know, we're all partners in this together. So we need to have everybody in there from the beginning so we can set the ground rules, figure out what we need to do, and then we'll really start pushing forward after that meeting to figure out what we need to do to optimize everything else, start evaluating a lot of these compounds in a number of mutations in some of those laboratory cell lines I mentioned. So I think 2 weeks from now will be an important meeting, but within the next year, I think we're really going to see a lot of this really shaping out based off of the outcome from that meeting.
Okay, great. So Jed, next question for you. So does this same process that you just told us about have the ability to affect or cure other diseases?
Yeah, very, very much so, yes. Induced pluripotent stem cells have the ability to go into any lineage that you can find in the body. So using gene editing techniques, this is active research that's going on in many different diseases. The monogenetic component of CF makes it very unique because we only have to correct that one gene. So it actually is an area where people are researching quite hotly, quite actively right now.
So we have two questions here, which are essentially going after the same idea. So are there efforts going on for CRISPR delivery into the lung? Do either one of you guys want to address that?
Sure. Yes, there are efforts that are looking at a multitude of different ways. So I introduced CRISPR and Cas technology to you guys today. This is kind of the most recent advance. People like it in the lab because it's really easy to use. But there are many other forms of gene editing that can be accomplished out there. So people are working on gene editing in the lung as we speak right now. One of the issues is what cells actually are going to be gobbling up those editing reagents. And when you think about stem cell biology, if you think of it in a form of evolution, that stem cell is actually hidden from the extracellular environment, from that environment, the lumen environment. So if you instill a virus into the lung, for instance, it usually gets gobbled up by the ciliated cell, and then that cell dies because you don't want the virus to infect the cell that's going to be causing the repair. So evolution has very smartly done that. But that's counterintuitive to us because if we want to use these known delivery methods, it's very difficult for us to get those delivery methods to a cell that's going to be a long-term benefit. So it's a little bit challenging, but the work is ongoing as we speak.
Okay, Catherine, next question for you. What needs to be done to determine a mutation's Thera type? Will this be a significant challenge when typing 1,700 different mutations?
And yes, it will be a challenge, but obviously there's people working in the lab every single day that are willing to work on this. But no, we've actually, one of the early stage efforts that we have is we're now trying to express quite a few hundred of these mutations that we think may be kind of the first ones that could benefit from these modulator therapies. So X mutations would probably not be high on the list, but the missense mutations where there may be a gating defect or a conductance defect, those are kind of being ranked a little bit higher, and we're going in and starting to do the studies to really understand how they may respond to these therapies. This is kind of the first stage, and those mutations that come out of this, I don't want to say screen, but this evaluation will then follow up with trying to collect patient cells and really understand if what we saw in the first cell models, if they will then carry over to the patient cells. So it's not something that's going to be done overnight. It's not going to be done in the first 6 months, but I think, as I mentioned, about the next year, we're going to have a better handle on some of these mutations and really start putting them into the buckets and understand where we need to put additional effort moving forward to really understand this and get the information out.
Okay, great. So let me jump back over to Jed. So you talked a lot about induced pluripotent stem cells, and you mentioned tissue-specific stem cells. There's a question here. Could cord blood stem cells also be used for the activities that you were describing?
So induced pluripotent stem cells, the only requirement you need to be able to make one of these IPS lines is that the cell is mitotically active or is capable of dividing. So really any cell type that can divide can undergo this process. And the beauty of this is we don't need to have banked down cord blood. We don't need to have that in the freezer. We can go and freshly isolate cells from anybody's blood, independent of whether or not they have these cells banked down. And you can use blood, you can use skin. They've done it from hair follicles. You can really, any cell that can divide can get reprogrammed.
So we have another question related to gene correction. With broad use of gene correction, are there other groups making headway as well? And is the CF lab working with any of those groups?
Yeah, we have quite. So I guess this is something that I should have been more clear on. This group of people that you see up here are the people that I work with every day. But in reality, the number of collaborations that we have out in the academic community as well as in the large pharma community and the startup community is unbelievable. We are in touch with many different people who are doing this. I don't know if I'm allowed to tell you who we're working with and all that kind of stuff. But yes, we are working with other pharma companies that are out there. Yes.
Okay, so this is a question from me, Catherine. So we talked about 1,700 mutations. That's a tremendous amount of work. Earlier, you put up a slide that showed the broad distribution of of the genotypes in the patient population. So can you tell us a little bit how perhaps the two different alleles or the two different copies of CFTR affect the amount of work that you expect that needs to be done for the Thera typing efforts?
Right, so it was a little misleading when I said, well, there's 1,700 mutations, and we've got 11 covered, and so we have 1,690 or 1,680 something left to go. It's not as bad as it sounds. So obviously, with the delta F508 homozygotes, those are taken care of. The gating mutations, those are taken care of. We've got that additional 4% that we think may benefit from modulators. That starts pulling out a piece of the pie. But there was about a 20 to 30%, I'm trying to think of the number, for the delta F508 heterozygotes. That will pull a large number of these mutations out as well. So if there are patients that have one of these rare mutations, but they have a delta F on the other side, if there are no patients that have a mutation that don't have the delta F, which that's probably a weird way to word it, we may not have to go through and evaluate those cell lines. So we're able to query the registry. We're able to find out which patients may fall into that last 5% category where they don't fall into the delta F or any of the other mutations that are currently approved. So the numbers will start to whittle down, but again, at the end, we'll be left with that 5% that is nonsense, and those that aren't going to benefit from the traditional modulator approaches that we're doing now. And that's why we have all these other initiatives underway for the one-time cure. Obviously, that would hopefully benefit everybody, but also, as we mentioned with the nonsense mutations, having specific screening programs to help address those patients as well.
I think we have 5 minutes left. Okay, so we have one really difficult question here, and I'll perhaps save that for the last one. Jed, back to you. How would you return modified autologous stem cells back into the airway?
This question came up in the previous session as well. Okay. Difficult question to answer, but one that there is, again, a little bit of feasibility in the studies that are going on right now. The first lab that really came out is Andy Vaughn's group. He works with Hal Chapman at the University of San Francisco. What they've been able to do is actually just put them down intratracheally or actually intranasal. So it's a very simple installation of those cells, but their research has very clearly shown that, yes, the lung is very good at getting things out. So you actually have to cause some sort of injury to the lung to be able to get those cells to stick. So the models that they actually have been using has been H1N1, which is that influenza virus everybody remembers from a few years back. It's a devastating virus, and they're doing this in the small animals, and that's actually showing engraftment and clonal growth in their models. But whether or not that's something that's really feasible for a CF patient, I can't recommend that we're going to go through H1N1 injury in a CF patient. But the feasibility, everybody, again, feasibility, it's there. These cells are engrafting and expanding.
So it sounds to me like, Jed, that one of the key things you actually need is space for this cell to actually set down and start growing.
Correct.
Without the space, we're just putting in cells, they're going to float around. They're either going to be killed or they're going to be cleared from the airway.
Correct.
So given the space, there's an opportunity for this term engraftment. Okay, so here's the very tough question. And so based on this technology, and I'm assuming that the technology that they're referring to, Jed, is from your presentation, how far off is the cure?
Can I defer this to Bill? I'm going to answer it this way, actually. Okay, so feasibility is a beautiful thing because it keeps us interested and it keeps us understanding that if we can just optimize and get better and optimize and get better, that there's a real chance of some good happening. But that doesn't give us any kind of timeline yet. Okay, and feasibility doesn't also mean that it's going to be the way that this will work. So I can't give a timeline, but what I can give you is the idea that we have this group that you see on that screen right there working every day, 7 days a week, 365 days a year on this. And we also have collaborations with academic groups all over the world that are helping us on this. This is not just my work. This is work of thousands of researchers. And so if we have a chance, we have a team that's built, that's working on this, and we're doing it as quickly as we can, but we can't do it too fast. We have to be systematic, we have to use empirical evidence, and that's how we're going to proceed. So yes, feasibility, awesome. I can't give a timeline because we're working on it.
Okay, thank you. So I think we've run out of time for today. Again, I want to thank Catherine and Jed for very nice presentations. And to all those that have submitted questions, thank you very much. Very insightful. Have a good night.